Hello everyone and welcome to my channel. Today discuss about plasma proteins. There are three plasma proteins. One is albumin, the next one is globulin and fibrinogen. The albumin and globulin is also known as serum albumin and serum globulin, and because fibrinogen is not present in serum, it is converted into fibrin during clot formation. Now the normal values of proteins are: total protein is 7.3 gram per deciliter and the range is 6 to 8 gram per deciliter. Albumin is 4.7 gram per deciliter, globulin 2.3 gram per deciliter and fibrinogen 0.3 gram per deciliter. Albumin by globulin ratio is normal in normal individual is 2.2:1. It is a good indicator for liver and kidney diseases. Molecular weight of albumin is 69,000 while that of globulin is 1,56,000 and fibrinogen is 4 lakh. Origin in embryo. Plasma proteins are formed from mesenchyme cells. First of all, the albumin is formed and later other plasma proteins are formed. In adults, mainly reticuloendothelial cells of liver is the site of production of plasma proteins. Other sites are bone marrow, spleen, disintegrating blood cells, and some gamma globulins are produced from B lymphocytes. Now there are certain functions which served by plasma proteins like coagulation of blood. Fibrinogen is responsible for the coagulation of blood. Defense mechanism: the globulins are responsible for mainly gamma globulins responsible for production of antibodies and protect our body from various diseases like influenza, other viral diseases. Transport mechanism: like hormones are transported by alpha and albumin and alpha and beta globulin, and the metallic ions are also transported by the globulins. Maintenance of osmotic pressure: many substances pass through the capillary membrane, but plasma proteins remain in the blood. Because of their size, they cannot pass through the capillaries, so they exert an osmotic pressure which is known as also colloidal oncotic pressure, which is 25 mmHg. Due to the pressure difference between the tissues and blood, various substances is passed through tissues from blood and blood from tissues due to Stirling's hypothesis. Another is regulation of acid-base balance. Plasma proteins are responsible for about 15% buffering capacity of blood, viscosity of blood. The albumin is mostly responsible for maintaining the viscosity of blood. Then other two proteins, erythrocyte sedimentation rate. Globulins and fibrinogen is responsible for the Rolex formation of RBCs. Rolex formation is mainly the piling up of the RBCs, and this Rolex formation is very essential for erythrocyte sedimentation rate, and this erythrocyte sedimentation rate is an important indicator for finding of many diseases. Suspension stability: the blood cells remain suspended in the blood during circulation, and this is due to the presence of fibrinogen and globulins. Triphone substances are actually responsible for the nourishment of tissues in culture, and these substances are produced from leukocytes, which are produced from the plasma proteins. Now, as reserve proteins, during starvation or inadequate protein intake or other situations when the protein level becomes low, then the plasma proteins converted into amino acids by tissue macrophages, and these are then carried by blood through those targets where it is needed. So by this way, these plasma proteins work as reserve proteins. Now there are certain conditions where there is decrease in protein levels, known as hyperproteinemia, when there is increase in protein levels, and hypoproteinemia, where there is decrease in protein levels. Hyperproteinemia is associated with acute nephritis or liver dysfunction, and there are also many causes like excess of glucocorticoids, and many other there are some other causes. And in case of hypoproteinemia, these are found in chronic infections, chronic nephritis, and cirrhosis of liver, diarrhea, pregnancy, malnutrition, etc. So this is a brief discussion about plasma proteins, and here we are ending our video. So see you in my next video, and if you like my video, please subscribe my channel.